All right, now that we've got our foldable made, let's flip it open and we're going to start with our mitochondria structure. I'm going to use a red pen for all of my labeling. All right, so this is a mitochondria. We're going to start. Mitochondria have two different membranes. So I'm going to start with this outside line over here. I'm going to draw a red line from the outside, and I'm going to label this my mitochondrial outer membrane. So it is the outer membrane of the mitochondria, but we are going to refer to it as the mitochondrial outer membrane. And just above, just on an area of kind of the snake looking thing, I'm going to write that that is the inner membrane, but we don't just call it the inner membrane. We call it the mitochondrial inner membrane. You can pause here if you need. I'm going to move it up just a little bit. All right, so now we're going to talk about these dots. So just pick like any three dots. I'm choosing these three here and connecting them. All of these dots are ribosomes. So ribosomes, remember, are our protein factories. Now we're going to label the white area. The white area is the intermembrane space. Inter means between, so it is the space between the two membranes. Then this loop that almost looks like a figure eight, this is our mitochondrial DNA. If we remember from our last unit of cells, mitochondrial DNA is one of our evidences that mitochondria used to be prokaryotic cells. Now, this time I'm pointing to just the space, not the dots with the white space. This is called the matrix or the mitochondrial matrix. And that is the fluid inside of the inner membrane. All right, these areas that I'm boxing in here, these are called the cristae, Cristae, and cristae are the folds of the mitochondrial inner membrane. They help increase the surface area so that more reactions can occur. The structure of the mitochondria, it's important that you know that each membrane, both the inner and outer mitochondrial membrane, is a phospholipid bilayer, and it has embedded proteins. The inner membrane itself is convoluted with enfoldings, and we call these things the cristae, and that the intermembrane space is very narrow, and that is the region between the inner and outer membranes. The mitochondrial matrix contains enzymes, ribosomes, and mitochondrial DNA. So we labeled both mitochondrial DNA and the ribosomes inside of the matrix. Why are these structures important? The foldings of the inner membrane, the cristae, they increase the surface area for cell respiration reactions, and the intermembrane space is small, so a hydrogen ion gradient forms very fast. All right, so we flip to the inside. I want you to notice that there are 6 circles. The molecule that we're starting out with is a hexagon shape. It is made of C6H12O6, which hopefully you know that molecule. Give you a second to think about it. That molecule's name is glucose. Now we're drawing those 6 carbons in a line. 6, I'm just putting a C in each one so that you know that each is a carbon. So we need to go and we need to input some energy. So we're going to start with ATP. Sorry about the error. And we're going to input that in. It's going to release a phosphate. I'm drawing a cloud burst because ATP has energy, and it's going to turn into a DP. And that's because the phosphate is going to attach to one side of the carbon chain. The same thing is going to happen on the other side. We're going to introduce another ATP. It's going to go into ADP because it's going to add a phosphate. These are still our 6 carbons. Now, This guy's name, his name is fructose 1,6-bisphosphate. You do not need to know that, but it's kind of neat because glucose and fructose are very similar, and the 1,6 means that the phosphate is on the 1 carbon and 6 carbon. But the next thing that happens is lysis, or the splitting. And so we now have 3 carbons and a phosphate on each side. This guy's name is G3P, or glyceraldehyde 3 phosphate. G3P is fine. And at the end here, we're going to go from G3P to a pyruvate. Pyruvate is also called pyruvic acid. I really don't care which one you call it. You just need to pick one name, but be okay that you recognize it. Now we have a G3P on each side, and we're going to end up with a pyruvate on each side. So how does that happen? Well, this time we're going to take an ADP and we're going to remove a phosphate, because notice that we do not have phosphate on the end. We're actually going to do that twice. So we're going to make 2 ATP, taking 1 G3P to a phosphate, and we're going to do that with each G3P. So we're going to end up making 4 ATP total. Now, in order to do that, we need to use our electron carriers. NAD plus is an electron carrier, and he's going to pick up an electron plus a hydrogen ion from our molecule. And that's going to turn him into NADH. Now, this probably doesn't make much sense. We're going to do some elaboration on this in just a few minutes. Pause if you need. All right, let's summarize the important parts of this glycolysis reaction. So if you remember at the beginning of the drawing, I started with introducing 2 ATP and giving their phosphates. So we want to make sure that we write that 2 ATP are needed to provide energy to start the reaction. And we make 4 ATP at the end. So what we say is that glycolysis has a net gain of ATP, that it is 2 ATP of as a net gain. So we also need to understand this concept of a redox reaction. Redox reactions are chemical reactions where electrons are moved or shuttled between molecules. There's 2 different parts of the redox reaction. There is the oxidizing, or the ox part, reactions, and these are the molecules that lose electrons. And then the reduction reactions, or the red ox out of the redox, are the molecules that gain electrons. Well, how do we even remember this? Well, if you remember the mnemonic device, oil rig, oxidation is losing electrons and reduction is gaining electrons. The last concept that you need to know is what an electron carrier is. So an electron carrier is a molecule that carries or shuttles electrons. So it could be the result of either an oxidizing reaction or a reduction reaction. Our example that I used in glycolysis is NADH can go to, or be broken down into, NAD plus 1 electron plus another electron plus a hydrogen ion. And that has the weird arrow because you can go back and forth. You can take those 4 products and they can become the reactants to form NADH. All right, let's learn about what happens during the Krebs cycle, which is also known as the citric acid cycle. So we ended up with pyruvate at the end of glycolysis. So we need to start with something called pyruvate oxidation. Remember, oxidation is oil rig, oxidation is losing electrons. So we're going to start off with our 3 carbon sugar, and we need to go to a 2 carbon molecule, and that 2 carbon molecule has a weird guy attached to him called CoA. So the first thing that we need to do, remember that we're going from a 3 carbon to a 2 carbon. So the first thing that we need to do is we need to take some electrons away. So we're going to have an NAD plus take 2 electrons and a hydrogen ion from the pyruvate. At the same time, coenzyme A or CoA is going to come in to the pyruvate and join with it, and we're going to release a carbon dioxide. So that's where our third carbon went to. Now we're ready to start the Krebs cycle or the citric acid cycle. So we're going to start here with 4 carbons. And all we're doing in this cycle is counting the carbons. So if you count the carbons over here at the next one, we now have 6 carbons. So 4 carbons to 6 carbons. Where are we getting the other 2? Well, we're going to get them from this molecule. Okay, so in order to do that, we're going to release the CoA, and then those 2 carbons that are left have now joined with the 4 carbons to form the 6 carbon molecule. Well, if we count the next set, now we have 5 carbons. So we need to release a carbon dioxide. In order to release a carbon dioxide, we're going to take an NAD plus, we're going to take 2 electrons and a hydrogen away, and then that's going to allow us to release a carbon in the form of carbon dioxide. And now we're repeating it because we're going from 5 carbons to 4 carbons. So again, we're going to take an NAD plus and an capturing 2 electrons and a hydrogen, and that's going to allow us to release a carbon dioxide. So now we're going from a 4 carbon to a 4 carbon, but we're going to do some other molecule rearranging. And in the process, we're going to create ATP, and then we're going to use another electron carrier, and that electron carrier is FADH2. So we're going to take an FAD, 2 electrons, and 2 hydrogen ions. And then we're going to form an NADH. All right, so it is important to know that all of this process, both the pyruvate oxidation and the citric acid cycle, this occurs in the mitochondrial matrix. So glycolysis occurred in the cytoplasm of the cell, and now we're in the mitochondria, but specifically in the matrix. All right, so what's our summary and our important points about the citric acid cycle? Well, it's really important to remember that carbon atoms are released from the citric acid cycle as carbon dioxide. It's also important to remember that FADH2 can be oxidized or reduced to form FAD with no charge plus a negatively charged electron plus another negatively charged electron and 2 hydrogen ions. So remember that H2 is really 2 H pluses. It's important to remember that FADH2 is another electron carrier. So we've already talked about 2 different electron carriers, NADH and FADH2. And you might be starting to feel like this is alphabet soup. It's also really important to remember that our starting molecule for the citric acid cycle is pyruvate. And as one pyruvate is broken down, we get 3 carbon dioxides, 4 NADHs, 1 ATP, and 1 FADH2. But we need to remember, how many pyruvates did we make from 1 glucose? Well, we made 2 pyruvates. So that means that it takes 2 cycles of the citric acid cycle are required for each glucose molecule. All right, before we get started actually labeling what goes on, we need to label some structure. So this is a lipid bilayer. I got a little lazy drawing on my phospholipids, but this is the outer mitochondrial membrane. Oops, I accidentally wrote matrix. It's the membrane, outer mitochondrial membrane. And then this guy here, this is the inner mitochondrial membrane. You can see the phospholipids over to the side. Again, I got a little lazy. This space in between is the intermembrane space. Now, in our first picture of the mitochondria, it was much, much bigger. It is actually a very small space, more closely shown here. All right, this space underneath the intermitochondrial membrane is the mitochondrial matrix. So remember, that's where our citric acid cycle happened. All of these blobs, these integral proteins, you don't need to know their numbers, but they are numbered 1, 2, 3, and 4. The little circle inside of 2 is called Q, and the big guy at the end is ATP synthase. All right, so we're going to start with one of our NADHs. That NADH could have been made from glycolysis or the citric acid cycle, and it's going to break off into some pieces. It's going to break off into NAD plus and H plus, and it's going to release its 2 electrons inside of complex 1. Those 2 electrons, are they going to move from complex 1 to the Q protein? And then the Q protein is going to take it to the little guy at the top. And then the little guy at the top is going to take it to the 4 protein. All right. As that happens, so that goes from there. Okay. So as that happens, when the electrons go into complex 1 is going to shoot some hydrogen ions into the intermembrane space. Then FADH2 that we just made in the citric acid cycle is going to release its 2 electrons also to Q and break off into FAD and 2 hydrogen ions. The 2 electrons that were released are going to go to Q. From Q is still going to take it to the little guy, and then Q is then going to drop them, or the little guy is going to drop them at 4. So now we have 4 electrons. As those electrons move through, complex 3 is going to spit 2 hydrogen ions into the intermembrane space. So now protein 4, now we have a buildup of hydrogen ions as that process is repeated for every FADH2 and every NADH we created. So that's important. But now we need something to capture those electrons. We're going to take 4 hydrogen ions and oxygen gas, and we're going to combine those with the 4 negative electrons to form 2 water molecules. So we say that water is the final electron acceptor. Because electrons are moving down the membrane, we call this an electron transport chain. So just this part is called an electron transport chain. So remember, we were pumping those hydrogen ions. So that's a tiny space that's becoming more and more positively charged. So we don't like that. So once it becomes too positively charged, ATP synthase will pull hydrogen ions through to balance and restore homeostasis. As it does that, that's going to power the bottom part, the rotor of ATP synthase, to join ADP plus a free phosphate ion to form ATP. This process is called chemiosmosis because we are restoring the chemical balance of the small space. The whole process, even though we shorten it and call it an electron transport chain, the whole process is actually called oxidative phosphorylation. So oxidation, oil rig, oxidation is losing. So we're losing electrons and picking them up to form ATP. Phosphorylation. All right, so what's our important information about the electron transport chain, or if you include all the process, it's actually oxidative phosphorylation. Well, we need to remember that NADH and FADH2, they are our electron carriers that are carrying the electrons to the electron transport chain or the inner mitochondrial membrane. We need to remember that oxygen is the final electron acceptor, and then it will form water. The intermembrane space is very small, even though neither of our drawings accurately reflect that. So the hydrogen ion concentration gradient is actually created very, very quickly. And that hydrogen ion gradient is actually what activates the ATP synthase to make the ATP, which is the whole point of cellular respiration. We also need to remember what our starting molecules are. We start with NADH that could have come from glycolysis or the citric acid cycle. We start with FADH2, which came from the citric acid cycle. And we start with oxygen, which is just one of our necessary reactants for aerobic cellular respiration. And we make two products. We make ATP and we make H2O. All right, so let's talk about fermentation. The first thing we need to know is that there's actually two different types of fermentation. There's alcoholic fermentation and there's lactic acid fermentation. All right, so it's important to remember that fermentation is the absence of oxygen. So we're going to start with glucose, okay? And they both start the same. They both start with that glucose, just like aerobic cellular respiration does. And alcoholic fermentation is going to end up with two pyruvates. So how do we get to two pyruvates? Well, we take 2 ADP and we make a net of 2 ATP, just like we did at the beginning of glycolysis. And when we do that, we're going to need to take 2 NAD plus and form 2 NADH. So it is similar to glycolysis. All right, so we have glycolysis happening. The lactic acid fermentation, it's the exact same thing. We go from glucose to 2-pyruvate. In order to do that, we're going to take our 2 ATP, ADP and form a net of 2 ATP. All of this is the exact same process of cellular respiration. We're going to take 2 NADH. We're going to take 2 NAD plus, and we're going to form 2 NADH. So again, this top part, it's the exact same. It's glycolysis. So what happens next, the second part, this is how we are different. In alcoholic fermentation, we're going to make 2 ethanol molecules. And in order to do that, we're going to release carbon dioxide. Well, how are we going to release carbon dioxide and form 2 ethanol? We're going to take 2 NADHs and we are going to reduce them, oxidize them, to form 2 NADHs. So where do we get these guys from? They are just shuttled back and forth. So the 2 NADHs that we made in glycolysis are going to be used in this alcoholic fermentation step. And then the 2 NAD pluses that we make are going to be shuttled back to glycolysis. So this is fermentation, alcoholic fermentation to be specific. And it is just the regeneration of NAD plus so that we can make more ATP when we get more glucose. It's really important to note that alcoholic fermentation occurs in yeast cells. All right, so what happens after we have those 2 pyruvate in lactic acid fermentation? Well, this time we're not going to make ethanol. We're going to make 2 lactate molecules, also known as lactic acid, which is how this fermentation gets its name. And this, we're not going to release any carbon dioxide. We're going to take 2 NADH electron carriers. We're going to release the electrons into the pyruvate to form lactate, forming 2 NAD pluses. And we're just going to shuttle those electron carriers and the electron molecules back and forth, just like we did for alcoholic fermentation. So this part is also just fermentation. And because its whole process is just an NAD regeneration. So the whole point is just to make a tiny little bit of ATP over and over again. In order to do that, we need to regenerate that NAD+. Now, there's lots of different cells that do lactic acid fermentation. Yogurt making bacteria, red blood cells do this because they don't have mitochondria, and our muscle cells do this when there is little to no oxygen. All right, so what are our key points about fermentation? Fermentation does not generate ATP, but it does regenerate NAD plus so that glycolysis can continue and we can continue to get those two net ATP. We also need to remember that there's two types of fermentation: alcoholic fermentation, which takes pyruvate and produces carbon dioxide and ethanol. And there's lactic acid fermentation, which takes pyruvate and produces lactic acid.